Good morning, and thank you for joining today's webinar, Considerations for Reducing and Refining Animal Research, Practical Advice for Adopting Human-Centric Models in Response to Recent Revisions to FDA and NIH Guidelines. Today, I am proud to introduce our speaker, Dr. Dwayne Dexter, whose career impressively spans both academic research, product development, and commercial leadership roles. Dwayne began his career as an associate professor at Penn State College of Medicine, focusing on cancer drug discovery, resistance mechanisms in breast and prostate cancer. Over the past 25 years, he has collaborated with scientific teams to develop advanced in vitro models and assays, including organoid bioprinting, organ-on-a-chip platforms, and disease-specific models such as chronic kidney disease and prostate cancer in underrepresented populations. Dr. Dexter has also played a key role in bridging breakthrough technologies to the research community, including high-throughput drug screening systems, high-content imaging, and single-cell isolation platforms. His unique combination of academic and industry experience gives him a valuable perspective on the practical adoption of human-centric research models. Please join me in welcoming Dr. Dexter.
Thank you, Melanie, for the introduction, and thank you, everyone, for taking time out of your day to join us on this interesting and timely topic. So for the agenda for today's talk, we're going to review some of the announcements that have come out of the NIH mainly. We'll review the drug discovery process. We'll talk about return on investment from our current approaches. We'll review the history of animals in research, particularly focusing on mouse models. Review the 3 R's in the ethical use of animals in research. We'll talk about the new approach methodologies, review some of those types of technologies. We'll talk about the challenges of utilizing those and qualifying those for drug discovery research. And then we'll discuss how Bruker preclinical imaging is supporting the reduction and refinement in the use of animals in research. So why am I here? Why are you here? There's been several announcements over the last several months from the FDA and NIH, specifically targeting the use of animals in research, and also highlighting and encouraging the replacement of animal models with NAMs such as organoids, tissue chips, AI, etc. Some of these announcements caught researchers off guard, but the director of NIH signaled this shift in his budget letter request to Congress in early spring, where he called out one of the initiatives and focus research initiatives of the NIH moving forward, essentially calling out the use of human-based or human-centric research technologies. This was further delineated and clarified in a workshop hosted by the FDA and NIH in early July, where Dr. Kleinstauer basically highlighted that we need to move away from using outdated animal models in our research, and highlighting the fact that moving forward, grant requests and grant proposals should include language on how one could use NAMs in their research. So if we look at a simplified overview of the drug discovery pipeline, it really hasn't changed too much over the last several decades. We make clinical observations of human health or disease. We reduce that observation into some easily manageable research unit, whether it's a cell, a protein, even a genomic piece of information. We look to identify targets, therapeutic interventions, identify compounds that would potentially act as a drug therapy. We look to evaluate these compounds in preclinical studies, typically using cell lines, mice, other methods. If everything goes well, we identify lead candidates that make it into clinical trials. If there's success in the clinical trial, the therapeutic agent is approved by the various regulatory bodies and it is commercialized. And then we have post-commercialization monitoring to make sure that there were no safety events that were missed in the broader population. So why do I talk about this? we often hear about the success of this approach to drug discovery. The number 90% drug failure or drug approval failure is bandied about. There's several ways that one can target or identify how that number is produced. There's a great study by Schumacher et al., where they looked at 18 pharma companies across about 14 years, looking at their drug discovery applications and the percent approval rate. So you can see, despite whether a company had, hundreds of INDs or, less than 50 INDs, the drug approval percentage hovered around about 14%. So a little better than the 10% that we often use as a benchmark. But I think this study, highlights quite well the success in drug approvals from IND. Another way of looking at this is on the right-hand side. Scannell et al. Published, I'd say, pretty interesting paper in 2012 looking at the return on investment in drug discovery versus approval rates, termed Eroom's law, a takeoff of Moore's law. You can see that, instead of doubling success rates, the more money that was applied into the drug discovery process, we actually had a lower return on investment. This data was updated in 2020. You can see that there's on the right-hand graph a modest improvement in return on investment, driven by a couple factors, the introduction of cell therapies, other biologics, and a change in the FDA approval process. But overall, you can see that, increasing investment in drug discovery has not yielded an improved return on drug approvals. Another way of looking at this information, another timely sort of review of investment, is if you look at the NIH budget over the last 20, 30 years or so. Even though NIH budgets have increased modestly or sometimes dramatically over this time frame, you can see with the blue dotted line, the number of INDs submitted really hasn't changed over this period of time, nor do you see a substantial shift in drug approvals. Academic institutions or not-for-profits also submit INDs and look for drug approval as well. But you can see that even though increasing NIH funding, we really don't move the needle on drug approvals either. So one of the things that we are looking at as a research community is, are we using the right models? Are we using the models correctly? And I think that's one of the impetuses of the NIH and FDA to look at alternatives to animal modeling. We've used animals or complex organisms for millennia to improve the human condition. And we've used a variety of these organisms in research, from the lowly worm all the way up to man, actually. So there are a variety of models that are used in research studies. Principally, some of the larger animal models, dog and non-human primates in particular, are mainly used in safety and PK/PD studies. The graph on the right actually shows an interesting trend, however. The United Kingdom has great reporting structures for the use of animals and how these animals are used in research. So I'm using their data as a surrogate, but I think generally these trends exist across the rest of the EU as well as in the United States. But you can see that since about 2014, you see a drop in the use or drop in procedures on animals over the last 10 years or so. So this is a good indication that we are finding better ways to use animals in research, or actually finding replacements. Again, using data out of the UK, you can see that the principal animal model used in research is mouse, followed by fish and rats. And why is that? Well, mice have been used for the better part of a century in research. They have a level of complexity that allows us to make some assumptions or translations into human biology. And these three models are actually easily genetically manipulated. They're easy to breed and create cohorts. So they continue to be a principal research model. Again, if you look on the graph on the left, you can see that there's been a pretty dramatic shift in the number of procedures, again, a surrogate for the number of animals used over the last uh, 15 years or so. But one interesting caveat here is you can see the use of genetically altered animals remains consistent over this time. So although the number of animals being used is reduced, you can see that the ratio of genetically altered to non-genetically altered is increasing. And this will be important later in the talk. So why mice? Mice have been of interest to humans for the thousands of years. Uh, you know, looking at the phenotypes of mice and collecting these interesting phenotypes. Obviously, we know now that these are the results of genetic mutations, and these have not only interesting phenotypes, but they also have interesting relationships to human biology. Mouse research really started to take off in the early 1900s, where William Castle and C.C. Little, who was working in Dr. Castle's lab, really started to use mice to understand genetics, mammalian genetics. This really started to take a foothold, not only at Harvard, but then started to expand across other research institutions once the Jackson Lab was founded in 1929. And you can see that some watershed moments for this early use of mouse in understanding mammalian genetics. George Snell and others won the Nobel Prize in 1980 for their early work in the 30s and 40s on identifying the major histocompatibility complex in mice and then translating that into human genetics. And then a watershed moment for mice in their use in research was in the early 80s when Palmeter et al. Produced the first transgenic mice expressing a human gene. So this really started a shift in how we thought about using mouse models in understanding human biology, and in particular human disease. So a couple of examples of some important mice, just to highlight some important discoveries on mice. So the famous OB mouse, the obese mouse, and variants of that has been instrumental in not only understanding appetite, bioenergetics, parabiosis. The leptin gene was cloned from this, and from that point, this mouse and variants of it have been used in understanding metabolism, but also interventions that could reverse, for instance, diabetes, and most recently GLP-1 and the different variants of GLP-1 inhibitors. More recently, humanized mice or human immune system mice have played a prominent role in the immuno-oncology space. There are different variants of these mice, but essentially these mice have mouse genes that have been knocked out to prevent the development and maturation of the human immune system. These mice then can be implanted with either primary human immune cells or stem cells, which will then produce functional human immune cells. There are various versions of this mouse, but it's played a prominent role in the development of immuno-oncology drugs and some of the cell therapies. As an example, here's a humanized mouse model where not only was the therapy addressed, but the actual disease, in this case, B-cell leukemia, uh, where this individual patient developed resistance to the CD19 traditional CAR T treatment, but it still expressed CD123. So this tumor was reimplanted back into one of these NSG or NRG type mice to reconstitute the tumor in the mouse model. So a humanized centric model of the tumor. And then these mice were treated with different therapies, either CAR T alone, CAR T19, CAR T123, a pooled treatment, or CAR T that targeted both moieties. So this is a great example of how this type of model is used to identify the appropriate therapy. You can see here that the dual targeting CAR had a complete response in one instance and a pretty good overall response in a second patient. These mice have also been instrumental in understanding a phenomenon called cytokine release syndrome, or is commonly referred to as cytokine storm. There have been a number of therapies that induce this type of systemic adverse event, uh, cell therapies, targeting agents, et cetera. As a great example of how this model can be used to assess adverse events, a famous case out of France was a molecule TGN-1412. It's a CD28 superagonist. Um, the cytokine storm activity was not detected in any preclinical analysis, but upon treatment of the first 6 patients in the phase 1, they all developed severe cases of cytokine storm. Retroactively going back now and using this CD28 superagonist, we can actually now replicate the clinical adverse events in this mouse model, seeing enlarged spleen, massive cell immune invasion in certain tissues. So a really interesting approach. Why this is important is because CD28 still continues to be an attractive target. So this type of model system can be used to make sure that the agonist or antagonist have had this cytokine storm activity engineered out. So a final example that I want to highlight is a real recent example is the case of baby KJ, which has been in the news recently. But this individual was born with a mutation on both alleles of the CSP1 gene. This protein is important for processing ammonia and converting it into the urea cycle. This baby KJ could not process this, and so researchers went about to identify the mutations and then identify possible approaches to actually repair the mutation in this baby. So you can see in a very short timeline that a CRISPR approach was identified. This was validated in a cell line that contained the mutation of baby KJ. And interestingly enough, a transgenic mouse model was made in parallel of the cell line, which A, exhibited some of the phenotype, but B, also acted as a model for the systemic delivery of the lipid nanoparticle, and can it target and repair the mutation within a complex system. Once this was proved, a clinical version of the lipid nanoparticle was developed, tested for immunogenicity and other safety profiles in a non-human primate, and again, a mouse. And within 8 months, baby KJ was treated through several rounds of doses of the lipid nanoparticle, and it's been shown that the mutation has been repaired on one allele in this individual, and baby KJ continues to be followed in terms of success and improvement in this particular mutation. So again, this is just a great example of how a complex in vivo system can be used to not only test the viability of a treatment, but also see if a phenotype can be repaired. So what are the 3 Rs? This is not a new phenomenon, even though we see quite a bit of about it in the press recently. But this was actually a ethical foundation established in 1959 by Russell and Birch, where we talk about the reduction, refinement, and replacement of animals in research. And just to highlight what each of these mean, reduction is obviously using less animals in a research protocol. Using smarter study design, for instance, using design of experiment to make sure that you are powering a study appropriately, but not overusing animals, making sure you're using the minimum number to power the study. taking advantage of new and improving methods, for instance, for Bruker, utilizing longitudinal imaging across a cohort of animals that are under study. That way that you're using the same animals over the life of the study as opposed to using multiple animals in endpoint analysis. And then also developing databases and repositories, which as we develop genomics data, other omics data, et cetera, these databases can be built and used to explore different types of phenotypes and genotypes, potentially limiting the number of animals or limiting the unintended use of a model that doesn't fit the appropriate disease. Refinement is, often refers to improved husbandry and yeah, how animals are handled, minimizing stress, improving enrichment environments to reduce psychological stress, using methods that are non-invasive so that we're trying to limit the amount of surgery, et cetera, on the animals. And of course, making sure that we're choosing the right model for the particular study. And then finally, replacement, which we'll talk about here in a little bit, is what are the alternatives to animal use? And of course, um, you know, the main push here is on NAMs or new approach methodologies. Some of these aren't so new, but organoids, organ on a chip, or sometimes referred to as microphysiological systems, 3D bioprinting, in silico methods, and more recently, machine learning, AI, computational methods. With all that said, I think we can't ignore two things. Biological complexity, by definition, is very complex, and there will be questions that will still require the use of complex living systems to address these questions. And also, I don't think we can ignore the fact that although we have this dismal return on investment in terms of drug approvals, we can't ignore the fact that there are tens of millions of dollars of drugs and diagnostics, et cetera, that are on the market that were approved that went through the traditional drug discovery process. So animals have played a critical role in the approval and commercialization of many drugs that have improved human health. So how do we get here in terms of NAMs? I just want to highlight, a few regulatory directives and other memorandum that have been out there. The EU has had a substantial head start in this space. They established a regulatory framework called REACH in 2007 with the intended goal of reducing the use of animals, implementing alternative methods in terms of hazard assessment or toxicity assessment of substances. This was further cemented in 2013, where they put a strict ban on the testing of cosmetic components on animals. And also in 2013, they passed a directive where they required strict reporting requirements on the use of animals. And again, encouraging and developing frameworks for the implementation and validation of alternatives to animals in research. So the EU has had a substantial, I'd say, focus and head start on this area, and it is reflected by a couple of things. Number one, if you look at the graph on the right, you can see that skin irritation, eye irritation tests before 2009 were mainly done in vivo, but due to the passage of the 2013 cosmetic regulation, you can see that there's been a substantial shift to in vitro methods for testing of these outcomes. I would say that although the EU has had a substantial, almost 20 years head start here, there still are a lot of in vitro model systems that are in development, but still haven't reached this level of use as we see with the skin and eye irritation test. If you want to understand how the EU is approaching the adoption and validation of NAMs, I just highlight one series on testing assessment 377. It's an interesting read if you want to understand the nuts and bolts of what goes into this type of evaluation. So this series is talking about developing in vitro tests for developmental neurotoxicity. So it's just a great example of the thought process that needs to go into the implementation and validation of a system that's going to replace one or two types of animal testing in terms of safety assessment. So in July, the workshop that was held, laid out some guidelines. There was, I would say, some robust conversation during this meeting. But essentially, the guidance from the NIH is that grant funding is no longer going to focus exclusively on funding grants that develop animal models only of human disease. Um, Grant funding requests are going to encourage researchers to explore the use of a wider range of models, particularly the NAMs. And if animal models are going to be part of the study, you really have to have a robust explanation for the use of the animal model, as well as, again, reiterating your commitment to the three R's. But with that said, is animal model research disappearing tomorrow? No, it's not. Traditional animal models, as I highlighted, say with the NSG mouse, continue to play an important role in our research studies. Our improvement and making these models human-centric or much more relatable to the human disease is also improving their use. So animal models will continue to be used here. But as scientists, I think we need to revisit our commitment to the three R's and how we're using these animal models or these cohorts in these preclinical studies, and making sure that we're taking maximum advantage of the use of these models and gaining the most efficient data from them. So I wanted to highlight a few examples of these NAMs. This concept is not new. As an example, I highlight two safety assays that were developed in the early 2000s. Phospholipidosis is one where cationic compounds tend to cause this adverse event or accumulation of lipids within a cell. Traditionally, this model was done in rodents, rats, where you would dose the animals with various concentrations of the compound, sacrifice animals at endpoints, do histopathology, and look for lipid accumulation. Very labor-intensive and costly approach here. Two different groups independently developed a high-content imaging assay where they were able to show that they could detect phospholipidosis. Both of these organizations, pharma companies, had a wealth of internal knowledge on compounds, positive knowns. So they were able to implement this type of assay to replace a very expensive rodent assay. A second example, Peters et al. Developed a cardiotoxicity assay using MEA or impedance to look for long QT syndrome and some other cardiotoxicity events. So in this case, you're using primary cardiomyocytes or iPSC-derived cardiomyocytes. You can monitor electrical activity across a time frame to see if your compound induces some of these classic cardiotoxicity adverse events. More recently, cell painting has been employed to look at not only compound efficacy, but also compound toxicity. Cell painting is a method, again, leveraging high content imaging, where you're staining multiple compartments within a cell, capturing images, segmenting and quantifying fluorescent signal from those compartments. Recently, a live cell painting reagent, ChromaLive, was brought to the market. This, unlike classic cell painting, which is an endpoint assay, ChromaLive allows you to do kinetic analysis or longitudinal imaging across a study. But both approaches collect a tremendous amount of imaging information, which then can be pushed through machine learning AI types of algorithms, where you can then start to develop fingerprints for, say, targets or pathways that might be of interest, or in the case of toxicity, looking for fingerprints that might predict, for instance, liver toxicity. So again, this is a more modern method that is starting to be employed across both efficacy studies as well as toxicity studies. But as you saw from the descriptions of NAMs, oftentimes these are referring to organ on a chip, organoid methods, 3D bioprinting. So as opposed to going into a deep dive of what these are, how to use them, I thought I would give a high-level overview of what's included, how these NAMs are developed. So first, I'd like to highlight the fact that stem cells and iPSCs have really changed a paradigm for how we do cellular research. We are able now to develop, I would like to call, patient specific models of disease. So for instance, isolating fibroblasts from a Parkinson's patient, using iPSC technology to create iPSCs, then differentiate those into a variety of different neural subtypes. And then using cell culture methods, we can do long-term studies looking for development of Parkinson-like phenotypes and then potentially drug interventions. We can also deploy CRISPR here, where we can start to repair mutations or induce mutations in these model systems. So a pretty powerful tool that is allowing us to do a better patient-centric research and moving away from some of the classic immortalized cell lines that we've traditionally used. Organoid methodology, it takes advantage of adult stem cells, a method developed by Hans Cleaver and his group out of the Hub. Basically taking adult differentiated tissue, employing organoid differentiation methodologies, where you can basically replicate, in particular, the epithelial components of a particular tissue or organ. So this is, again, another powerful method where we can take disease or normal tissue from an individual and replicate the cellular components from that individual. And both the iPSC stem cell and organoid approach can then also be deployed in the organ on a chip 3D bioprinting space. These methodologies now start to build on this notion that we need complexity in our system to really truly understand how our biology is behaving, how our molecules perform in a complex system. And so these tools really employ the development of complex co-culture systems. You can see on the left-hand side the cartoon of an organ on a chip. These often employ perfusion methods where you can perfuse in medium, drug compounds, antibodies, cell therapies, etc. Shear stress seems to be important for the development of some complex systems. It seems to be critically important for the development of endothelial tubes. So there are a number of organ on a chip systems that are out there. I just highlighted a couple here. And of course, 3D bioprinting is also another approach. There are 3D bioprinting methods where you could print into an organ on a chip, or you can print into microtiter plates. One of the advantages of 3D bioprinting is the fact that you can now start to add another level of complexity by controlling the matrix environment that your cells are in. So matrix is a key driver of a number of physiological events, for instance, fibrosis. So being able to modulate both the peptide or matrix components as well as the stiffness, again, adds another level of complexity to the assay. So now if we start to layer in NAMs in our research workflow, we can see that NAMs are going to play and are playing a critical role across the early and preclinical phases of research. So developing, as I said, patient-centric cell lines or disease-specific cell lines or organoids, is improving upon the, we'll call it efficacy of drug response. These model systems can also be used to develop better in vivo models, for instance, using organoid methodology to develop patient-derived organoids in the tumor space. We now can really rapidly develop patient or personalized diagnostic, if you will, tools that can then be used in vivo in the animal model studies. So these methods are currently being employed across the early phases of research. I just want to highlight a couple of examples here. Um, Liver toxicity, as you recall, I mentioned drug failure rates. Um, Although oftentimes people think of drug failure, drug approval rates due to safety, but that's actually a modest reason for failure in the clinical trial. But if drug safety is one of the reasons, liver toxicity is one of the principal toxic events that cause withdrawal from clinical trials or even post-approval. So I just want to highlight three examples of how NAMs are being used to develop in vitro liver toxicity assays that both contain the complexity of the liver, but also start to allow us to explore liver toxicity in different ways. So on the far left is a study from Bursack et al. Using the mimetis organoplate. And again, this allows for a higher throughput assessment. And what's interesting from this paper is highlight two findings from this paper is one, they used iPSC-derived hepatocytes, which is an interesting use of the hepatocytes here because these are often have fetal-like phenotypes, but putting these into an organ-on-a-chip system under perfusion, you actually differentiate these hepatocytes into a more adult-like phenotype. So using a cell source that's repeatable and reproducible is one finding from this paper. The other finding is that dose response was critical in identifying compounds that cause drug-induced liver injury. So making sure that you're able to do a dose response and finding that window where drug-induced liver toxicity was found. On the far right is a recent paper from Inspiro and the National Center for Toxicology, and they used a spheroid-like approach here, again in 96 or 384 well plate, again, so high throughput screening. It had a very high positive predictive value. But what's interesting is it had a very high predictive value for compounds that were targeting the nervous system. So an interesting little caveat within this study is that it had a very high predictive value for a particular class of compounds. And the center study is a study from Emulate, where they used their organ on a chip. And again, two principal findings from this paper is it had, again, a very high predictive value in terms of detecting compounds that were known liver toxicants. But the other interesting thing is Scannell was also part of this paper, did an economic analysis. And if the liver on a chip method was deployed before IND and clinical trials, the compounds that were identified here would not have gone into clinical trial, and these organizations would have saved about $3 billion. So an interesting economic analysis on the use of these technologies. Uh, So one of the questions that continues to be brought up, not only in NAMs but in general in our drug discovery process, is how can we address diversity earlier on in our research programs, in mainly the in vitro preclinical space. So one approach is to take advantage of the iPSC methodologies or organoid methodologies and start to build massive biobanks of individual contributors. and use a brute force approach to study these and screen these. That's one way. Another clever way is village in a dish. If you're not familiar with that, I point you to the paper by Powell from Joseph Powell's group here, where you take advantage of iPSC methodologies, and I suspect you could probably do organoids at this in this approach as well. But you take advantage of the iPSC technology where you combine individual iPSCs across donors into a single dish, differentiate those into the cell type you're interested in, do the treatment or intervention, and do your analysis and use deconvolution to identify the contributors to the particular phenotype or that you're looking for. So a clever idea of how one could look at diversity, but do it in an efficient manner. In vivo, we can also look at diversity. This is a great example, using the diversity outbred strain from the Jackson Laboratory, where you take a variety of inbred strains, cross them against each other, and you develop a single colony that has genetic diversity, but genetic diversity that you can easily track. So it's a clever idea of how to use in vivo tools to replicate diversity within a population. And these diversity outbred colonies can be used to study a wide variety of activities. They've been used to study genetic factors that influence cardiotoxicity. They're being used to understand how aging impacts, for instance, loss of immune cell components as we age. So it's an interesting approach of using mice and developing genetic diversity that is easily tracked. I mean, that's the key thing here. There have been some interesting proposals of how we can now layer on human-centric mutations in these types of mouse colonies so that we now start to use the genetic diversity to understand why, for instance, certain individuals have modest sickle cell crisis versus others that will have a deep sickle cell crisis. As I said earlier, the EU has had substantial head start in this space. The NIH is obviously requiring the consideration of NAMs and other methodologies. So, as opposed to us having an entire seminar on how to use these and build these types of models, I just want to highlight there are a number of great resources that are out there already that you can find publications, working groups, et cetera, where you can access a number of resources to understand how these tools are being utilized, what models are currently being developed, calls for funding, calls for partnerships. The foundation for NIH, the second one down there, actually has a call out for public-private partnerships to start the validation and qualification a process for interesting organ-on-a-chip or other NAM methods. So I reference those as well, that you can find a lot of useful information. In terms of machine learning, AI, this is not a new phenomenon. Computational methods, for instance, have been used for more than a decade to improve upon lead or hit identified compounds, to improve upon their physiochemical properties, to even identify efficient synthesis pathways, and then upon different testing methods, circling back and even refining the methods. So computational methods in terms of drug discovery is not a new concept. What is new is how to use machine learning and artificial intelligence in the drug discovery process, but also in the disease or target identification process. Again, using the UK and sort of their approach to the 100,000 Genome Project, which has been expanded now to proteomics and real-world clinical data. We are collecting large amounts of or libraries of information across patients, cell lines, et cetera. So there's a vast amount of information, often disparate information, that we can utilize in terms of understanding disease pathways, potential targets, new targets, et cetera. So Machine learning AI is, I mean, there's several companies that have been founded based on this approach, is becoming an important tool here. And so I just sketched out a rough, pathway of how AI is used, where we're combining these large libraries of data, looking at the clinical situation, and using machine learning to identify targets or potentially compounds. For instance, PROTACs are often designed using machine learning AI. If you're not familiar with PROTACs, these are molecules that promote the degradation of proteins within a cell. These outputs then, in theory, could be pushed through another sort of in silico AI method where in silico experimentation could be done, where we can look at various outputs from compounds or therapeutics that could potentially identify tox signals, evaluate pharmacokinetics, look at off-target effects, and hypothetically even identify a treatment that could potentially be developed completely in silico and never touch a cell or a mouse. So um, pay attention to this space, because as these tools, in particular, the data sets that are employed in these tools improve and grow, this will become an important component of not only the drug discovery process, but clinical practice. As an example, I chose this example because it's combining a couple of different NAMs here that we just talked about. So this is a recent paper from Hesperos where they developed a we'll call it multi-tissue organ on a chip that contain hepatocytes, splenocytes, and HUVEC cells to look at malaria and drug response in malaria. And I won't go into a deep dive in the paper, but using the in vitro organ on a chip method, they were able to develop some pharmacokinetic profiles on dosing and response, and using AI and coupled with clinical data, you're able to actually generate a digital twin now from a patient for a patient, and potentially identify a personalized dosing regime for that patient. So an interesting approach where they're combining multiple NAM methods here to improve upon treatment for patients. What are the challenges here for adoption of NAMs? I would say first, the NAM community has been very, I'd say, a niche type of research area. I do see more and more publications appearing, but if we want this to become a broadly utilized approach, we're going to need to change our approach to education and really start to introduce these concepts earlier in scientists' research career. We need to continue to develop successful use cases, be transparent in those use cases. If using liver toxicity, for instance, there's a validation trial that's starting using multiple liver chip or other NAM approaches to identify the best ones or a combination of ones that could improve upon detection of liver toxicity outside of an animal approach, and continue to, on the education side, offer training and workshops, either the commercial providers or funded through various private entities or the NIH itself. Again, encourage collaboration. The success of these new tools is only going to happen if we have cross-sector partnerships and collaboration. By that, I mean pharma company and biotech companies working closely with academics and other not-for-profits that are exploring the development and validation of these. But the pharma sector really has the tool sets and some of the internal knowledge to really validate and qualify these things. The regulatory environment, obviously, as we start to build out standards, making sure that these are communicated appropriately, sharing submissions that utilize these tools, I think will be important. Not sure how that will work in terms of transparency. Continuing to generate open access databases. There are already a few that are out there. And then, of course, funding. Funding is going to be super critical here. A recent editorial by Dr. Wafik El-Deary, the head of Brown Cancer, talking about the recent NIH announcements, talked about how these models are expensive and and probably won't reduce the cost of drug discovery. While that's true, I don't think that's the right way to look at costs. I refer back to the Emulate paper, where if liver toxicity was detected, over $3 billion would have been saved. I also think we need to think about it from the perspective of the patient. Is like think about how many patients were inadvertently put into clinical trials where efficacy was not going to be achieved, or we actually worsen their health status by causing some unintended adverse events. So I think looking at the cost of these, there's actual, the cost of using the technology, but I think we need to look at the long-term cost as well. So, yes, this is a statement from August 15th. There's a lot of fast-moving communication occurring from the NIH. I just wanted to highlight a director statement to the NIH community. Two interesting comments within his statement is he referred to the publish and perish culture, and I think that's a great comment from him in that we need to start to change our culture of how we're doing research, come back to the basics of why we do research. Ultimately, it's to improve the human condition. So I think we need to think about that in terms of how we move forward with our research. And so that's interesting call out by the director. The other is that there is going to be an office established, the Office of Research, Innovation, Validation, and Application. Again, coming back to resource. So pay attention to that as they come out with guidelines, hopefully tools and resources to better educate the community on how to successfully navigate this new grant requests. But what was also interesting is that you see a slight softening of language here where that strategic initiative to develop, et cetera, NAMs to complement animal models. So I think that's an interesting comment there that we're sort of moving away from we need to stop using animals to complementing animal model research. With that said, this isn't going to happen overnight. The mouse has a 100-year head start on this space, and I think you can see from the EU, even though that they've encouraged and supported implementation for the last almost 20 years, progress has been slow in the NAM space. So how is Bruker working to support reduction and refinement? Here's just an example of, coming back to Dr. El-Deary's comment about costs. As we move to smarter animal models, I'll just highlight, the NSG mouse. I just did a quick publication search over the last 14 years. And you can see the rapid, almost doubling adoption of the use of NSG and humanized NSG mouse in publications. The reason I highlight that is these advanced mouse models or human-centric models tend to be 10 to 100 times more in cost than an inbred strain. So even though NAMs are expensive, so are these new models that we're employing in terms of mouse research. So one of the things that Bruker is doing is, we're making sure that we continue to educate our end users in the community on the proper methods and detection tools that can be used in using these models. Again, another resource, I talk about proper models. This is just a great example of, Black 6 mouse was developed, by C.C. Little and, almost 100 years ago. So is that the right mouse to use for understanding metabolic diseases? In some cases, yes. In a number of cases, no. So making sure you choose the right model, even though it might be slightly more, in the long term will be more beneficial to your research. Again, a couple resources on how to identify right disease phenotype models, the phenome database at JAX and the systemsgenetics.org database as well. So Bruker preclinical imaging, we have a strong emphasis on reducing and refining the use of animal models. Bruker preclinical imaging has the largest or broadest portfolio of preclinical imaging technologies. And because of this, we could take advantage of our internal expertise across the different modalities. And we're developing and building tools to allow not only longitudinal imaging, but multimodal imaging. So where one can use one readout, I'll talk principally about bioluminescence and fluorescence here in a second, but using early detection methods to identify disease progression, then using something that has a much higher resolution or a different type of readout to further understand the disease. So why longitudinal imaging? Here's just an example on the bottom. And I did this in my research where you figured out how many animals you needed for your study. You treated these animals, and at some time point in the future, you sacrificed a number of animals to do histopathology, understand, in my case, tumor response. Then at a second time point down the road, you would sacrifice another set of animals. And this was not only labor intensive but costly. You're using a lot of animals. With longitudinal imaging, in this case using bioluminescence, you can use the exact same cohort of mice across the entire length of the study, and utilize each individual animal in the cohort as a control unto itself. So this not only is a powerful approach, but it also starts to address the reduction in use of animals. And again, highlighting modality, making sure that you're choosing the right approach and right modality is also critical here. So again, Bruker has expertise and experience across all the modalities that are pictured here. So we definitely have a lot of experts to help you identify the right starting points and the right research study design to take advantage of your cohort of animals. So how does animal imaging fit into in the broad spectrum of the drug discovery workflow? So obviously I'm highlighting preclinical imaging here. So you do your in vitro analysis, your organ on a chip analysis, and what would be the next step? And the obvious logical next step that many researchers move to is using bioluminescence or fluorescence readouts. Why is that? Well, one, this is the most sensitive method that is used to detect biology. Two, developing bioluminescence or fluorescent readouts or reporters, you utilize the standard laboratory methods. So the methods to developing these cell types is not uncommon to most folks doing modern day molecular and cell biology. The other advantage is that although you can do early detection here, you can run longitudinal studies. The development of BLI, FLI does not interfere with downstream analysis. In particular, you can go into other preclinical imaging modalities, or you can use these animals for endpoint studies. For instance, doing spatial proteomics like nanostring, you can then look at tumor response, look at biological activity, recover the material and run spatial proteomics on that material and compare it back to the actual clinical situation. So this approach really allows you to extend the use of these models, not just across the imaging study, but also into other areas of readouts. Again, statistical power, something that I've mentioned several times. I will say to the audience here, making sure that you are really addressing and understanding the statistical power of your study design, so you're not overusing animals. Longitudinal studies reduce inter-animal variability because the animal is its control unto itself. You reduce sample size. Longitudinal imaging is incredibly sensitive in terms of BLI and FLI. And of course, temporal resolution, looking at response over time as opposed to doing a single snapshot. This allows you to understand, for instance, does my therapy have an initial response rate, but then the tumor has a bounce back? Just a couple of examples here, looking at T cell leukemia. this study was done with 20 mice. So you can see that cohorts of 5 were used across a longitudinal imaging study. We were able to generate survival curves here and look at various treatment regimes to see which one provided the best response. Again, another example of where multimodal imaging and combined with bioluminescence is important. In this case, in terms of refinement of your study, this is an intratibial injection of osteosarcoma. You can see in the mouse in the right-hand side, we've got intertibial expression of the osteosarcoma, but you can see in the mouse in the upper left there that it's just outside the tibia. So what this demonstrates is that moving the mice forward in the study, we know that the mouse on the left is not the appropriate model to use, and we could remove that particular mouse from the preclinical study. It's also an educational moment here, making sure that the staff are trained appropriately and understand what proper delivery of the cell of interest into the tissue or compartment of interest is. And again, a last takeaway is again using BLI to evaluate early time points. So in this case, it's orthotopic delivery of A549 cells into the lung and looking at how successful we were with the orthotopic delivery and then the tumor take rate. So you can see that we can detect within hours of orthotopic injection of the A549 cells. So again, it just gives you an example of the sensitivity and early detection. And then these mice were subsequently kept on study for a number of weeks where other imaging modalities were used. But you can see that within days, we can use bioluminescence and fluorescence, whereas MRI or micro-CT takes several weeks. Following up on the director's statement on complementing animal models and how NAMs complement, NAMs are definitely going to find a place and use in sort of the traditional in vitro drug discovery space, understanding lead compound performance, making sure disease phenotypes are established. But in the preclinical space, NAMs are also going to play an important role and complement animal model research as well. NAMs potentially can be used to figure out dosing regimes that would go into an animal model. There are definitely areas where NAMs are going to be a preferred approach because animal models are not very informative for human disease or toxicity. An example is male reproductive tox is one. In terms of drug discovery and efficacy, female reproductive models aren't so great for in the animals. So just two areas where NAMs will probably definitely start to gain traction over animal models or improve upon animal model research. So with that, I'd like to just wrap up and summarize. Animal models continue to have a place in our research for foreseeable future. Um, you will definitely start to see a shift towards more human-centric types of models, and I'm definitely a fan of this. But we also need to continue to educate and encourage smarter study design and the use of longitudinal studies, whether it's longitudinal imaging or using imaging coupled with other endpoint readouts to maximize the use of these cohort of mice. NAMs have shown great promise in certain areas. I highlighted liver tox, which is definitely an important area where I think these organ on a chip or other NAM methods will have critical value. Some of the areas that we need to focus in on is lack of standardization, and that's both in protocols, but also how do we industrialize NAMs? There are a lot of organ on chips that are produced within individual research labs, but standardization requires industrialization. There are several commercial companies that exist already. I highlighted a few of those, but developing high quality tools, reagents, plates, et cetera, is going to be critical to making sure that these technologies are adopted and reproducible. And lastly, education is going to be an important resource, and for the folks that have control of the purse strings out there, in order for us to advance the mission and the guidance here, we're going to need to educate, especially in the early careers of scientists, continued proper use of mouse models, how to use these across longitudinal studies, making sure that study power is appropriate, improving non-invasive probes and readouts. There's just a recent paper on developing a new F-18 probe on looking at fibrosis. So, radiopharmaceuticals, radiodiagnostics continue to be used. Actually, radiopharmaceuticals are making a comeback. So, animal models are going to be critical in the evaluation of these molecules. Again, I talked about study power and then continued publication and transparency in the NAM space. So I want to thank you for your time and attention, and I'd be happy to answer any questions.